Here, due to lack of chlorophyll, this shows heterotrophic mode of nutrition. Right? What is the study of fungi? Study of fungi nothing but mycology. Study of algae is phycology, while the study of fungi is mycology. Right? Who is regarded as father of mycology? Myceli. Father of mycology is mycology. Mycology means study of fungi. Who is regarded as father of mycology? Father of mycology is Myceli. Is considered as father of mycology. Here, father of modern mycology is Dibari. Is also considered as father of plant pathology. Pathology means to study the various diseases of plants which are caused by different organisms. That is called as the pathology. So, father of modern mycology and father of plant pathology is Dibari. Right. E.J. Butler. E.J. Butler is regarded as father of Indian mycology. Father of Indian mycology. E.J. Butler is considered as father of Indian mycology. Okay, now let us discuss about characters of fungi. Here, generally the plant body of algae is thallus. Here, the plant body of fungi is called as mycelium. Plant body, plant body. The plant body of fungi is called as mycelium. Here, the mycelium is composed of a long, thin, thread-like structures which are called as the hyphae. Okay, the hyphae are intervening with each other, then form mycelium. Okay, here the mycelium nothing but plant body of fungi. Just it is composed of hyphae, right? Okay, here generally yeast. Here, except yeast, all fungi are multicellular. But here, the plant body of yeast is unicellular. That's why that plant body do not appears as mycelium. Hence, the plant body of yeast is called as pseudomycelium. Why does the plant body of yeast is considered as pseudomycelium? Because generally, all fungi are multicellular except yeast. Yeast is unicellular fungi. That's why the plant body of yeast is known as pseudomycelium. Generalizer, the plant body of fungi is mycelium. But what is the pseudomycelium? Where it is seen means yeast. Here, the plant body of yeast. Yeast is only unicellular fungi. Here, the plant body of yeast is called pseudomycelium. Plant body of yeast is called pseudomycelium. Right. Cell structure, generally the cell wall. Here cell wall of algae generally is composed of cellulose, hemicellulose, pectin, glucagons, galactans and mannans. Here, but here, what is the nature of cell wall of fungi? Generally, the cell wall of fungi is composed of chitin. Chitinous cell wall is characteristic feature of fungi. Here, fungi cell wall is not only composed of chitin. Here, it consisting along with the chitin polysaccharides also polysaccharides. So, the fungal cell wall is composed of not only chitin, it is composed of polysaccharides also. So, chitin and polysaccharides both are the components of cell wall of fungi. Cell wall of fungi. Right here. What do you know about chitin? Is it complex polysaccharide or is it complex polysaccharide or simple polysaccharide? Means it is complex polysaccharide. Here, whether it is homopolysaccharide or heteropolysaccharide, here the chitin, the cell wall material of fungi is chitin. Here, chitin is complex polysaccharide. Okay, right. Here, whether it is homopolysaccharide or heteropolysaccharide, absolutely it is homopolysaccharide, but not heteropolysaccharide. It is very important. Please remember this. Here, chitin is homopolysaccharide. Right. Here, what is the reserve food material of fungi? Here, generally, the reserve food material of fungi is glycogen and oils. Glycogen and oil. Glycogen and oils are reserve food materials of fungi. Yeah, just this is the cell structure of the fungi. Here, right. Generally, here the fungi are preferred to grow in. Most of the fungi are preferred to grow in humid and warm places. Here, very important point. Here most of the fungi are they prefer to grow in warm and humid places. Okay, right. Here what is the habitat, what is the nature, here what is the nutrition of the fungi? Here being heterotrophs, which does not consisting with chloroplast. That's why most of the fungi are parasites. Next, some fungi are saprophytes and some fungi are symbionts. Right. Here, the parasitic fungi. Generally, the parasitic fungi can cause diseases to the some plants. Here, saprophytic fungi. The saprophytic fungi here growing on dead and decaying of organisms. Here, they consume the food materials from dead and decaying organisms. Next, some fungi shows symbiotic association with lichens and roots of higher plants. Here, let us see first about parasites. Here, these parasitic fungi can cause diseases to the plants. Here, what are the examples of parasitic fungi? Here, first one is albugo, albugo. Here, it causes white spots on mustard plant. Mustard plant. Nothing but which can cause to white spots on the cruciferae. Mustard belongs to Brassicaceae. It is also called cruciferaceae. Here, second one is Phytophthora. Phytophthora. It is another parasitic fungi which can cause early. blight of potato. Sorry, not early blight. Sorry. Late blight of potato. Late blight of potato. Phytophthora can cause late blight of potato, which is also called Irish famine. Right. Another fungi, Cercospora personata and Cercospora arachidicola. Here Cercospora personata. Here which can cause ticka disease of groundnut. Ticka disease of groundnut caused by Cercospora personata and Cercospora rachidicola. Next, this belongs to Deuteromycetes. Next, Alternaria. Alternaria solani. Here it causes early blight of potato. Phytophthora causes late blight of potato. Alternaria solani causes early blight of potato. Next, Plasmopara. Here which cause downy mildew in grapes. Downy mildew in grapes or grapevine. Next, Colitotrichum palcatum. Colitotrichum palcatum. It is also a member of Deuteromycetina, Mycotina. Here which can cause red rot of sugarcane. Next, Helminthosporium voriae, leaf spot on rice. Helminthosporium voriae, here leaf spot on rice. Next, Eustylagocyte homine. Eustylagocyte homine. Here it causes smut disease in sorghum. Smut of sorghum. Next, Coccinia. Rust of wheat. Next, Trichoderma polysporum, which functions as against of root-borne pathogens. Next, Rhizopus, which can cause contamination of bread, hence it is called bread mold. Here, which appears as pin-like structure, that's why it is called pin mold. Rhizopus or called bread mold appears as pin-like structure, that's why it is called pin mold. It is also called black mold. Here at the time of maturation or at the time of asexual reproduction, which releases black color spores. That's why it is called as black mold also. Here these are somewhat parasitic fungi. Okay. Next, some fungi are showing symbiotic association with algae, which is called lichen. Then some of the fungi, these are symbiotic association with the roots of higher plants. Those are called as the mycorrhiza. Here now, let us see about symbiotic fungi. Here lichen. Here lichen is a plant body which is composed of two components, algal component and fungal component. The algal component is called phycobiont. Fungal component is called mycobiont. Right. Here, most of the phycobionts belongs to green algae. Mycobiont belongs to members of Ascomycetes and members of Basidiomycetes. Here, what are the functions of phycobiont and mycobiont in the component of lichen? Here, generally the phycobiont shows photosynthetic nature, which prepare the food materials. Those food materials are provided to the fungal partner. Here, the mycobiont provide the water and minerals to their algal component, and they provide shelter for the algal component also. Here, which is called this a type of symbiotic association does exist between algal component and the fungal component, which is called as the lichen. Here, generally, the lichens are serves as good pollution indicators. Here, the sulfur dioxide pollution, these are very sensitive for sulfur dioxide pollution. Here, in presence of pollution, the lichens cannot grow in presence of pollution. Here, my question here, A is an organism. A is an organism which is more preferable to grow in polluted areas. B is an another organism which is not preferable to grow in polluted areas. Here, what are the A and B? Here, listen carefully the question. A is an organism which prefer to grow in polluted areas. B is an another organism which is not preferred to grow in the polluted areas. What are the A and B respectively? Okay, I think you know the answer. Here, what is the correct answer? What is the A and what is the B? Here, A nothing but cyanobacteria. Blue-green algae. Here, blue-green algae form blooms in the polluted water bodies which are called algal blooms. So, cyanobacteria are preferred to grow in polluted water bodies, but lichens do not grow in the polluted areas. Those are very sensitive to pollution. Which are sensitive to pollution means lichens are sensitive to pollution. Okay, here in which pollution means sulfur dioxide pollution. Now this is about the lichen. Okay, here according to Whitaker's classification, here the lichens are cannot kept any kingdom. He cannot classify. So Whitaker's classification system is the five kingdom classification system. Okay, here the lichens cannot kept in the five kingdom classification system by Whitaker. Okay, viroids, prions, and lichens. These are not classified. These are not put in the five kingdom classification. But here among the virus, lichens, viroids and prions. Only lichens are the cellular, except all are the non-cellular. Okay, here the cellular organisms, which is not classified by the Linnaeus. The cellular organism which was not classified by the Whittaker means lichen. The acellular forms which are not classified by Whittaker's means virus or viroids or prions. Here listen carefully. The cellular organisms which are not classified by Whittaker's is lichen. Cellular organisms which are not classified by Whittaker means virus, viroids, and prions. All those are the acellular. So some acellular forms and cellular forms which are not classified by Whittaker. Okay, this is about the like it. Right. Another symbiotic association. Another symbiotic association is mycorrhiza. What is the mycorrhiza? Myco means fungi. Rhiza means roots. Here the fungus they show symbiotic association with the roots of higher plants which is called as the mycorrhiza. Those roots are called as the mycorrhizal roots. Okay, here what is the best example for mycorrhizal roots? The suitable example, the best example for mycorrhizal roots are pinus. Mycorrhizal roots are cycas. Mycorrhizal roots are seen in the pinus. Then mycorrhizal roots are seen in the cycas. Okay, right. Pinus. Pinus is gymnosperm. Here it shows mycorrhiza. Okay, mycorrhizal roots are seen in the finest. Here now this is about the parasitic this is about parasitic fungi, saprophytic, and symbiotic fungi. Okay, right. Now we are going to discuss about the reproduction of fungi. Okay, right. Here generally the fungi reproduce by vegetatively, asexually, and sexually. Here first of all vegetative reproduction. Vegetative reproduction. Here asexual reproduction. Third one is sexual reproduction. Here first of all let us see about vegetative reproduction. Here the fungi reproduce by means of vegetatively by fragmentation. What is mean by fragmentation? Here the fungi reproduce vegetatively by means of fragmentation. Second one is budding. Budding and fragmentation both are belongs to vegetative reproduction. Here right, what is vegetative reproduction? What is fragmentation? What is fragmentation? What is vegetative reproduction? Nothing but here vegetative reproduction does not involve formation and fusion of gametes like here like asexual reproduction. Okay, so vegetative propagation is also a type of asexual reproduction. Why? Because in the asexual reproduction and vegetative reproduction common criteria, there is no formation and there is no fusion of gametes. Right? Yeah. The fungi reproduce vegetatively by means of both fragmentation and budding. What is mean by fragmentation? The body of organism is split into many pieces. Each piece is called as a fragment. Generally, every fragment is developed into new individual. That is called fragmentation. Second one is budding. Here, generally, the budding is seen in unicellular fungi, which is called as the yeast. What is mean by budding? Budding is a type of cell division. Here, whether it is mitosis or amitosis or meiosis. Here, budding nothing but just this is the yeast body. Here, the protoplasm of the yeast come out from the body, then form a bird-like structure. Then form a bird-like structure. Here, now each bird is develops into new individual. Here sometimes the chain of birds are formed on the east. Sometimes the chain of birds are formed on the east. Here now this chain of birds is called as the torula. Torula. This stage is called as the torula stage. That's why the east shows torula stage. Hence that east is called as the torula east. What is the torula stage and what is the torula east? What is the torula stage? The torula stage nothing but a chain of birds remain present on the parental body after their formation. Such a stage is called as the torula stage. Here now that yeast is called as the torula yeast. Okay, right here. Now the budding. Here, whether the budding is mitosis or amitosis or meiosis, budding is nothing but amitosis, but not mitosis. Here, mitosis is the equatorial division, but here unequal buds are formed here. Okay, remember here, yeast shows budding. The buds nothing but these are equal or unequal means unequal budding. Here, this is not equal. Unequal buds are found. That's why this is the cell division. It is nothing but it is not a mitosis. It is absolutely a mitotic division, unequal division. Here, whether the spindle formation take place or does not take place during the budding. Here, generally during the mitosis, the spindle formation will be take place. But here in the budding, whether the spindle formation take place or will not be take place means the spindle formation take place during the budding, but that spindle is symmetric spindle or asymmetric spindle, equal spindle or unequal spindle. Here, budding is a unequal division. That's why the spindle formation take place during the budding is unequal spindle or asymmetric spindle. This is the very important point here. Is shows budding. Budding is not a mitotic division, nothing but unequal division. Here, which type of spindle is formed? A symmetric spindle or unequal spindle is formed during the budding. Here, very important. Here, I think you got the clarity about the budding, right? Here, spindle unequal or unequal spindle or asymmetric spindle is formed during the budding. Here, budding is unequal division. Unequal division. Okay, right. Let us move another type of reproduction. Here, the another type of reproduction is asexual reproduction. Here, generally in the fungi, the asexual reproduction. Here, asexual reproduction is carried out by formation of spores. Formation of spores. Here, these spores, ploidy of spores are haploid. Here, these spores, they may be different types of spores are there. Motile spores called zoospores. Non-motile spores are called aplanospores. And another type of spores are conidiospores. Conidiospores. Here, asexual reproduction, which is carried out by different types of spores. Here, generally the ploidy of spores is haploid. Here, these are the different types of spores are produced by different fungi, belongs to different classes. Here, right, first of all, zoospores. Here, what are the zoospores? The flagellated or motile spores are called as the zoospores. Here, zoospores also called as the planospores. Zoospores also called as the planospores, which are produced from zoosporangium. Okay, second one is the aplanospores. Aplanospores, nothing but non-motile spores are called as the aplanospores. Here then conidiospores. The conidiospores are produced in the ascomycetes. Here generally here the conidiospores. Here now just this is a conidiophore which is produced from basal part. Here just this is called conidiophore. Here now this conidiophore produces bulged structures. These bulged structures are called as the medullae. Medullae. Here now this is conidiophore. Conidiophore. Here it produces a bulged structures. These are called as medullae. Here the medullae produces a bottle shaped structures. Bottle shaped structures. Here these are called as the sterigmata. Sterigmata. Here now these pterygmata is able to produce a spores. These spores are called as the conidiospores. Here it is very important. Conidiospores. Here the conidiospores produced from conidiophore. The conidiophore develops a bulged structures called as the medullae. The medullae again produces or develops a bottle shaped structures. These bottle shaped structures called as the sterigmata. These sterigmata are able to produce as conidiospores. Conidiospores. So the origin of conidiospores, whether it is exogenous or endogenous. The conidiospores are asexual spores which are produced from conidiophore by exogenous origin, but it is not endogenous origin. Here zoospores are produced within the zoosporangium, that is the endogenous. Zoospores are endogenous spores, conidiospores are exogenous spores. This is very important. Here asexual reproduction carried out by spores. Those spores are flagellated called zoospores. Zoospores are produced within the zoosporangium. Okay, here within the zoosporangium, when the walls of zoosporangium are break, now the zoospores are released from that zoosporangium. So the origin of zoospores are endogenous origin. Remember, this is very important here. Applanospores also produced from within the sporangium. That's why zoospores and applanospores both are endogenous origin, while the conidiospores are exogenous origin. Exogenous asexual spores are conidiospores. Endogenous asexual spores are zoospores. Okay, yeah, just this is about the asexual reproduction carried out by different fungi. Okay, now we will go about we will go to sexual reproduction. Okay, here generally the sexual reproduction in different algae, which includes there are three events are there in their sexual cycle: plasmogamy, karyogamy, and meiosis. Okay, here plasmogamy, karyogamy, and meiosis, these are occurs in all classes of fungi except fungi imperfecti. Fungi imperfecti nothing but belongs to deuteromycetes. Plasmogamy, karyogamy, and meiosis never take place in deuteromycetes. Hence, those are called as the fungal imperfecti or imperfecti fungi. Okay, here, right. Here, in the sexual reproduction of fungi. Okay, here plasmogamy. Here plasmogamy, karyogamy, and meiosis. All these three events are immediately occurs in the all these three events are immediately occurs in the phycomycetes. Here, in case of higher fungi such as ascomycetes and basidiomycetes, these are belongs to higher fungi. Here in these higher fungi, here plasmogamy will take place. Here now the karyogamy does not immediately occurs after plasmogamy. Okay, hence the dikaryotic phase is formed, which is seen in the higher fungi such as ascomycetes and basidiomycetes. Okay, here what I said here, the sexual reproduction in the fungi involves various stages, sequence of events. First one is the plasmogamy, then after karyogamy, then after meiosis. Here all these three events does not occurs in the deuteromycetes. That's why those are called as the imperfect fungi, or also called fungi imperfecti. Okay, right. Then we are discussing about phycomycetes, ascomycetes, and deuteromycetes. In the phycomycetes, let us see here. In the phycomycetes, what I am saying here, listen carefully. Right. In the phycomycetes, here, plasmogamy, karyogamy, and meiosis. All these three events are immediately take place. But here, in case of ascomycetes and basidiomycetes, the karyogamy will not occur immediately after plasmogamy. Okay, right. What is the plasmogamy? What is the karyogamy? And what is the meiosis? And where it will be take place? Now let us discuss. First of all, sexual cycle. sexual reproduction. Here now, first one is plasmogamy. Plasmogamy. Here plasmogamy means fusion of protoplasm between two different strains of mycelium. Here in the heterothallic species. Heterothallic species means both male and female are different. Here those are differentiated into plus and minus strains in case of fungi. Here now that is called as the heterothallism. Heterothallism, which was discovered by A.F. Blacksley in Rhizopus. Right. What is plasmogamy? Plasmogamy nothing but fusion of protoplasm. Fusion of protoplasm. Here after plasmogamy, what will be happens? Karyogamy. Here what is mean by karyogamy? Here just this is the one mycelium belongs to plus strain. Here this is the another mycelium belongs to minus strain. Here both are having with protoplasm. Here nucleus also a part of protoplasm. Here right here both protoplasm are fused. Here protoplasm is fused, that is called as the plasmogamy. Here plasmogamy after carried out by karyogamy is followed after plasmogamy means fusion of nucleus. Here now these are the two different strains, okay, come together with each other, and the walls between the two hyphae are dissolved by the action of an enzyme called chitinase enzyme. Okay, chitinase enzyme. Here both are closely associated with each other. Then after their association, the chitinase enzyme is released by the fungi. Here now which dissolves the walls. Here now the two protoplasms are fused. Two protoplasms are fused. Here just this is the fusion of protoplasm. Here after that this nucleus and this nucleus, the plus and minus, the nucleus of plus and minus strains are fused. Now this is called as the karyogamy. Karyogamy. Here now after karyogamy, which is formed here 2n, nucleus belongs to 2n. Here it is haploid, it is haploid. After karyogamy it becomes to diploid. Okay, here what I am saying here, plus strain is haploid, minus strain is haploid. After karyogamy means after fusion of both of the nucleus belongs to, it shows diploid cat. Here now after that the meiosis will be take place. Okay, here generally these are the sequence events of sexual reproduction of the fungi. Here which is all these three events are immediately occurs in only phycomycetes, but not in the ascomycetes and but not in the basidiomycetes. These three events will never take place in the deuteromycetes. Here now let us see about the sexual reproduction of ascomycetes and basidiomycetes. Right here now, first of all, plasmogamy will take place. Here, after plasmogamy, the karyogamy, there is a gap between the plasmogamy and karyogamy. The gap between the plasmogamy and karyogamy is more. Here, what I am saying here, the gap between the plasmogamy and karyogamy is not take place in phycomycetes. The gap between the plasmogamy and karyogamy is more in case of ascomycetes and basidiomycetes. Here plasmogamy means fusion of two mycelium, fusion of two primary myceliums. Then plasmogamy takes place. Here after plasmogamy, delayed karyogamy. Delayed karyogamy. Here now protoplasm is fused. Here it has one nucleus, it has also one nucleus. Now this is called as the dikaryotic phase. Dikaryotic phase. Here the dikaryotic phase is generally haploid. Dikaryotic phase is generally haploid, binucleate, and it is saprophyte. Here now this dikaryotic phase develops into a mycelium that is called as the dikaryotic mycelium, or also called secondary mycelium. Dikaryotic mycelium, or also called secondary mycelium. The dikaryotic phase generally produces a mycelium called dikaryotic mycelium, or also called secondary mycelium. Now this dikaryotic mycelium or secondary mycelium produces fruiting bodies. These fruiting bodies are ascocarp and basidiocarp. Fruiting body of ascomycetes is ascocarp, and fruiting body of basidiomycetes is called basidiocarp. Here now the ascocarp contains a sac-like structure called ascus. Basidiocarp has a club-shaped structure called basidium. So the meiosis, the karyogamy, and meiosis both are take place in the ascus and basidium respectively. This is about sexual cycle of ascomycetes and basidiomycetes. Okay, plasmogamy here means fusion of two primary mycelium. Here now after plasmogamy generally karyogamy will be take place, but in case of ascomycetes and basidiomycetes not take place, it is delayed. That's why a dikaryotic phase is formed, that is called dikaryotic mycelium. Generally, the dikaryotic phase is very important haploid, binucleate and saprophytic stage. Now this dikaryotic phase is develops into mycelium called dikaryotic mycelium, or also called secondary mycelium. This dikaryotic mycelium or secondary mycelium produces fruiting bodies. These fruiting bodies in ascomycetes called ascocarp, basidiomycetes is called basidiocarp. Here ascocarp consisting with a sac-like structure called ascus, basidiocarp is consisting with a club-shaped structure called basidium. Here the karyogamy and meiosis both are occurs in the ascus and basidium respectively. Okay, in case of ascomycetes and basidiomycetes, this is about the sexual reproduction of fungi. Okay, now classification of fungi. Here, what is which characters are mainly considered? Which characters are mainly considered while classifying the fungi? Here, generally, the classification of fungi into four classes: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. It is mainly depends on the number one. Uh, Mycelium, structure of mycelium. Second one, mode of spore formation. Mode of spore formation. Okay, based on the nature of mycelium. Okay, based on the mode of spore formation, the fungi are classified into four classes: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Here, first of all, Phycomycetes. Phycomycetes. Phycomycetes are generally called algal fungi. Why these are considered as algal fungi? Because of their aquatic habitats. Phycomycetes are generally considered as algal fungi because of their aquatic habitat. Here, these Phycomycetes members are the mycelium of these. Mycelium is branched and aseptate. Here generally this is the hyphae. Here now this hyphae here it is consisting with multinucleated mass of protoplasm. Here without any cross walls, that is called as the aseptates. That's why cenocytic condition is happen. Cenocytic condition. Here seno means group, group of nucleus. Many nucleus are present in only one mycelium. Here which was it is formed as a result of there is no presence of any cross wall in that hyphae. Here mycelium is branched and accepted. That's why xenocytic condition. Xenocytic hyphae, that's why it is called as xenocytic hyphae, because branched and accepted. Here the every cell has the protoplasm and every cell has the nucleus. Here generally those cells are separated by crosswalls, that is called as the septa. In lack of septa, due to lack of septa, here all nucleus are appears in a only one cell, that is called as the xenocytic condition. Example, rhizophus and mucar. Rhizophus and mucar. Here next, asexual reproduction. Asexual reproduction is carried out by formation of zoospores. Here these asexual spores are exogenous origin. Sorry, here zoospores are always endogenous origin. Endogenous origin. Here next, sexual reproduction. Here already I said sexual reproduction, plasmogamy, karyogamy and meiosis. Here, then after karyogamy, meiosis will be take place. Then, as a result of sexual reproduction, spore is formed. That spore is called as the oospore. Pleidia of oospore are also called. It is also called zygospore. Plasmogamy and after karyogamy, the spore is formed. That is called as the zygospore, or also called as the oospore. Here, oospore or zygospore is formed as a result of sexual reproduction. Here, which undergoes into meiotic division. Now, this produces haploid spores. These haploid spores are developed into haploid mycelium. This is about the sexual reproduction in the phycomycetes. Okay, examples of phycomycetes: albugo, Phytophthora, Rhizopus, Mucor. All these are belongs to Phycomycetes. Second one is the Ascomycetes. Here, Ascomycetes. Ascomycetes. Here, these are called as the sac fungi. Here, these members of Ascomycetes are saprophytes mostly. Here some are present on the, some are saprophytes, some are they are present on the dung also. Here which are called as the coprophillus. Here these Ascomycetes members includes penicillium, yeast. Here penicillium. Yeast is especially useful for preparation of baking used in the baking industry. It is used to preparation of biscuits and used to preparation of breads. That's why that is called as the baker's yeast. Here, that same yeast is especially useful for preparation of beverage industries. Hence, that's why that is called the brewer's yeast or wine yeast. Penicillium. Here, Penicillium chrysogenum and Penicillium notatum. Here, these are especially responsible for production of a wonder drug. That wonder drug is called the penicillin. Penicillin is a antibiotic, first antibiotic which was discovered by Alexander Fleming. Here, it is synthesized from Penicillium chrysogenum and Penicillium notatum. Both are belongs to Ascomycetes. Here, next, Neurospora. Neurospora. Here it is extensively used in genetic work. That's why Neurospora is called as Drosophila of plant kingdom. Here Drosophila also used in genetic work. But here Neurospora it is called Drosophila of plant kingdom. Why? Because it is used very extensively in the genetic works. Hence it's called Drosophila of plant kingdom. Next, Aspergillus. Here just it is common weed of laboratory. That's why it is called as the guinea pig of plant kingdom. Guinea pig of plant kingdom, Aspergillus. Okay, here these are the some members of Ascomycetes. Now let us see about the mycelium. Here the mycelium of Ascomycetes is branched and septate. Branched and septate. It does not consisting with cenocytic hyphae. Okay. Asexual reproduction. Conidiospores. Conidiospores are exogenous. Next, sexual reproduction. During the sexual reproduction, they produces us fruiting bodies. Already I said, those fruiting bodies are called ascocarp. Here, these three types of ascocarps are there: Clistothecium, Perithecium, and Apothecium. Perithecium and Apothecium. Clistothecium, globe-like ascocarp without apical opening called Clistothecium. Perithecium, flask shape ascocarp. Apothecium, cup or saucer shape ascocarp is called apothecium. Here, these ascocarp consisting with a sac-like structure called ascus. Ascus. Here, the meiosis does take place in the ascus after karyogamy. Here, these ascocarp, all these are the ascocarp. Here, which consisting with the sac-like structure called ascus. The karyogamy take place in the ascus. Then after meiosis, division also take place in the ascus. As a result of meiosis, the spores are produced. These spores are called as the ascospores. Here, conidiospores are asexual spores. Ascospores are sexual spores. Asexual spores are produced exogenously. Asexual spores are produced exogenously. Sexual spores are produced endogenously. Very, very, very important. Here, ascospores. Pleidia of ascospore, haploid ascospores, endogenous. Conidiospores are exogenous. This is about Ascomycetes. Now let us see about Basidiomycetes. Here which includes rust fungi, smut fungi, bracket fungi, next puff balls. All these belongs to Basidiomycetes. Here these members of Basidiomycetes, the mycelium, aseptate and branch, no cenocytic hyphae. Next, asexual reproduction absent. Why? Because there is no asexual spores. Next, sex organs also absent. Here, the sexual cycle involves plasmogamy, karyogamy, and meiosis. Here, they form fruiting bodies called basidiocarp. Basidiocarp has club-shaped structure called basidium. Here, karyogamy and meiosis both are occurs in the basidium. Then, after meiosis, they produces spores called basidiospores. Basidiospores. Sexual spores of Ascomycetes, ascospores, which are endogenous. Sexual spores of Basidiomycetes are basidiospores. These are exogenous. Here it is very, very important. Asexual spores in Phycomycetes, endogenous. Asexual spores in Ascomycetes, exogenous conidiospores. Asexual spores in Basidiomycetes, absent. Asexual spores in Deuteromycetes also conidiospores, exogenous, right? Then sexual reproduction, sexual spores, oospores, zygospore that is diploid, that is in phycomystis. Here ascospores are sexual spores which are produced in ascomystis, that is endogenous. Ascospores endogenously produced sexual spores. Basidiospores exogenously produced sexual spores. Next, deuteromystis, sexual reproduction is completely absent. Here this is basidiomystis. Now last one is deuteromycetes. Deuteromycetes is called fungi imperfecti. Why those are called fungi imperfecti means there is no sexual reproduction in these deuteromycetes members. Here the asexual reproduction is carried out by conidiospores which are exogenous spores. Here in the deuteromycetes, after discovery of a particular sexual stage in the deuteromycetes, those are belong to kept in the other classes such as ascomycetes and basidiomycetes. Okay, the deuteromycetes members already we said some parasites trichoderma, then Cercospora personata, Arachidicola, Cultotrichum palcatum, Alternaria solani, Gibberella fusicorea also, bacterial disease of rice. All these are belongs to deuteromycetes. Here these members of deuteromycetes are involved in the nutrient recycling, nutrient cycling. Here this is very important. This is deuteromycetes. Here six kingdom classification system. After that, here six kingdom classification system proposed by Carl Hoss. Here, according to him, those six kingdoms are bacteria, archaea, protista, fungi, plantae, and animalia. All these are the six kingdoms. Among these six kingdoms, prokaryotic kingdoms are two. Then four are eukaryotic kingdoms. Okay. Here he placed bacteria and monera in place of bacteria and archaea in place of monera of Whittaker's classification system. Whittaker's classification system is five kingdoms, but Karl classification system is two kingdoms. What are the two kingdoms are additionally included by Karl means bacteria and archaea. What are the common kingdoms of Whittaker's and Karl are protista, fungi, plant and animalia. These are the common kingdoms. Okay, here what is the difference between the classification of Karl and Whittaker? Instead of Monera, the Karl proposed bacteria and archaea. Here, all these three kingdoms are arranged as a cluster of three categories. Those are called as the domains. What are the three domains? Bacteria, archaea, and eukarya. Bacteria, archaea, and eukarya are domains. What are the three domains? Bacteria, archaea, and eukarya. Eukarya. Here, bacteria is prokaryotic domain, archaea and eukarya. Bacteria and archaea both are the prokaryotic domains. Eukarya is eukaryotic domain. Prokaryotic to eukaryotic kingdoms, so 2 is to 4 means 1 is to 2. Prokaryotic domains to eukaryotic domains, 2 is to 1. Here, all these three domains are originated from common ancestor or common progenitor. Here, in these three domains, archaea and eukarya are closely related with each other comparatively to bacteria. Okay, children, the portion of biological classification is, I think, completed. Tomorrow we will discuss about discuss about another topic. Okay, children, read carefully. Have a nice day. Thank you for watching. See you tomorrow.
వేరే బ్యాచెస్ రైట్ రైట్ థ్యాంక్ యూ
ఇప్పుడు మీదేనా సార్ క్లాస్
వేరే బ్యాచ్ ఫిజిక్స్ రైట్ గురుజీ థ్యాంక్ యూ
అక్కడ నుంచి చెప్తారా
ఇంటి దగ్గర నుంచి చెప్తారా
రైట్ సార్
రైట్ రైట్ రైట్